Hello, everyone. This is to welcome you to the 42nd ESID Grand Rounds webinar. Today's topic are somatic mutations. Somatic mutations we identify in our patients suffering from inborn errors of immunity. And I'm very pleased that 4 great speakers have agreed to talk about this. But before I introduce the speakers, I would like to lay out some rules. First of all, and most importantly, it is not allowed to record the webinar. We will be doing this from the ESID side. So the ESID will be recording, and you can then on the ESID website later on follow up on the webinar if you were not able to listen in person today, or if you want to listen to it again. But please do not use any automatic transcriber or automatic note takers because it's not allowed to document by yourself. Myself, I'm Bodo Grimbacher. I work in Freiburg as a clinician scientist. I'm in the field of inborn errors of immunity when it was still named primary immunodeficiencies. So that is starting in the early 2000s. And since I've been working in the genetics identification of these patients, and therefore I'm very interested to hear and learn much more about most recent developments in the field of somatic mutations. For that topic, we have invited 4 speakers. I will introduce them one by one. The talks will be given sequentially, and you will not have chances to ask questions after each talk, but we will have ample time to discuss your questions after all the 4 talks have been given. Please put your questions into the Q&A and not into the chat. Please put your questions in the Q&A session you find below so that we can collect them and I can moderate the Q&A discussion after all talks have been given. Without further delay, I would like to introduce Dr. Cooper. Megan Cooper is Chief of the Division in Pediatric Rheumatology and Immunology and Professor of Pathology and Immunology at the Washington University in St. Louis. She cares for patients with pediatric rheumatology and immunology in her clinic, and her interest is especially on immune dysregulation. Her laboratory, however, focuses also on basic mechanisms of immune cell activation and immune cell regulation, using genetic sequencing to uncover novel molecular mechanisms of pediatric disease, including her most recent work on STAT3, for instance, on TLR8. So, Megan, thank you so much for accepting our invitation to talk today about the work you have been doing and in general about somatic mutations we identify or you have identified in your patients. Take it away.
All right. Thank you, Bodo. It's a pleasure to be here, and I'm very excited for this entire session and looking forward to learning a lot. So I'm going to start really with a bit of an overview of thinking about mosaic variants and how we think about them in inborn errors of immunity, or you'll hear me use the term genetic errors of immunity because these are not always inherited variants. As we kind of move forward with this webinar and dive deep later into some of the specific diseases. So just to get everyone on the same page by what we mean when we're talking about genetic mosaicism. And so genetic mosaicism is the concept that not every cell in our body has the same genetic code. And for example, you could have parents who are seemingly healthy. They do not have a pathogenic variant either in their germline or in their somatic cells or in their germ cells, for example, in ovaries or testes. However, that child has a genetic variant that is disease-causing in a subset of their cells. And so that is genetic mosaicism in this child. And this can occur at any time during development. And so we often think of somatic mutations occurring later in life with age or being associated with malignancies, and that is certainly true, and that can lead to mosaic GEI. However, these mutational events can also occur early during development. And one concept for me that has been interesting to think about, especially being a pediatrician, is the fact that our genome is not fixed. And so I think that we all realize this, that with every cellular division, there's a chance to acquire new mutations. However, the estimates are from these two very nice papers that came out of the Sanger Institute that when we are born, about 0.2% of the genome of our hematopoietic stem cells, which are obviously the cells that give rise to our immune system, is already mutated. So at birth, our immune system already has a genetic variation. Each year, we acquire substitutions that can lead to amino acid changes. And each year, we also acquire a lower number of insertions, deletions in our genome. And so they did this by looking at single nucleotide variants in clones of hematopoietic progenitor cells. And then later on, they actually looked at the mutational rate in HSCs and in circulating granulocytes. And interestingly, most of the genetic variation seems to come at the HSC level rather than the fully differentiated cell, because they saw a similar rate of mutations in HSC as in granulocytes. So, why is this relevant for us as rheumatologists? And so I'm going to tell you about a case here. This was a case that was led by Dr. Erica Schmidt in our group. And this was a 7-year-old girl who had CNS demyelinating disease leading to polyneuropathy. She had quite a bit of difficulty walking. She had also an autoimmune adrenal insufficiency. So this is a patient that would come into our hospital with difficulty ambulating. She would require high-dose steroids for improvement. Her clinical genetic testing was quite interesting. So we had sent a panel gene test, one of those 500 gene panels, that demonstrated a variant of uncertain significance in STAT5B, shown here. At the same time, she was also seen by our genetics colleagues who sent a clinical exome that was non-diagnostic. And actually, the exome did not report out the same STAT5B variant as the panel test. So the STAT5B variant, though, was of interest. It was previously reported as being somatic in cancer, and previous studies had suggested that it led to gain of function of STAT5B. And as immunologists, we know that Mosaic gain of function variants in STAT5B can lead to immune dysregulation, typically more eosinophilia and not associated necessarily with the phenotype that we saw in our patient. However, we suspected that this could be a variant of that disorder. So testing of patient cells, shown here on the left, a healthy versus patient, stimulated with IL-2, show that there is hyperphosphorylation of STAT5B in the patient cells. And then a reporter assay done in Josh Milner's lab showed that the patient's variant, when stimulated, acted similar to another well-known gain-of-function variant in STAT5B that we sometimes see in STAT5B somatic disorders. But it was still puzzling to us that this variant was not reported on her exome sequencing. And so it turns out in this case, the panel testing was done on a blood sample, whereas the exome sequencing was done with saliva. And so this was a good reminder for us that it really matters what cells you are sequencing. And their starting point of what you're sequencing is going to give you the results of potential variants in that particular sample. So we went on to look to see what cells actually harbor this STAT5B variant. And so we used droplet digital PCR, which is a quantitative PCR method where DNA samples are put into 20,000 or so different oil droplets. There's a little reaction that occurs in each, and you can say whether each droplet has reference sequence or has the variant sequence. And so what we see is if we look at the patient's whole blood DNA, we can see that it's about 50-50, either the STAT5B variant that we think is gain of function versus reference. However, we got a skin biopsy from this patient and generated a fibroblast line, and we can see that it's not detected at all in her skin fibroblasts. If we look at the variant allele frequency, and I think we're going to hear a little bit more about some of this terminology in Laura's talk as we think about how do we actually detect these variants. But we look at basically the percentage of reads that have the variant of interest, the variant in STAT5B. We can see that it's about 50% in all of her hematopoietic cell types. So if we look at monocytes, T cells, NK cells, whole blood, and then in the bone marrow, it's also at 50%. We had a buccal swab from the patient, and we can see a little bit of a signal of the variant there. And it's important to remember that buccal swabs or saliva, while they are enriched for epithelial cells, do generally contain at least about 20% immune cells. And so you are going to get a little sampling of immune cells there with those tests. It was absent from fibroblasts, as I showed you from the patient, and absent from the blood of the parents. And so this is an example where the variant occurred in a hematopoietic progenitor. It actually likely occurred very early during development, probably within the first 4 to 6 weeks of embryonic development, because it seems to have affected the entire hematopoietic compartment. And so because we see the variant occurring in all different cells of the immune system, we know that this is an earlier variant. For example, if a variant occurs later, for example, like in a common lymphoid progenitor, it may be limited to a specific cell type, such as in this case, showing all lymphocytes, or it could occur even later in occurring in a very specific cell population. And so this diagnosis of a somatic genetic error of immunity in this patient due to STAT5B, knowing that it was limited to her hematopoietic compartment, really led to a change in care for this patient with this demyelinating disease. And she underwent hematopoietic stem cell transplantation. And so we can see here by ddPCR after transplantation, so before about 50-50, after transplantation, there's a little bit of a signal showing that there's a little bit of residual host cells there, but she's now almost 3 years out of transplantation, in remission, has a normal gait. And so this is just an example to illustrate how identifying these mosaic variants in our patients can affect clinical care. The other topic that I wanted to address today is, as a clinician and as a physician scientist, trying to think about, okay, if a patient has a mosaic variant, how can it actually lead to disease? And so we think about this in a couple of different ways. And so if you have a genetic variant, I think that a couple of things must be true for it to affect the function of a cell. It should be expressed in a relevant cell type. So it should be expressed in an immune cell. It should be expressed in a relevant immune cell for the disease that the patient has. And it should have some type of an impact on the protein. And so you saw in that prior case that it was a missense variant that altered the function of the protein. You could have an early stop codon. You could have a variant that affects splicing. You could have a variant in a promoter region. So there's many different ways that variants could have an impact on a protein. And then there's multiple different genetic mechanisms by which a mosaic variant or a variant present only in a subset of cells could actually influence the immune response. So there have been several examples where a secondary hit in that cell type that has the genetic variant, it acts as a driver mutation and expands those immune cells that have the genetic variant, therefore bringing out the phenotype. You could have loss of heterozygosity, where you already have a inborn or a germline variant in the same gene, and you have a second hit as a somatic variant. You could have co-occurring variants on the same allele, and they could influence each other. And we can sometimes see this loss of heterozygosity or co-occurring variants, for example, in somatic FAS, for those variants in those families where there is a lot of incomplete penetrance, and much of that is due to a second hit in the same gene. And then from very nice work from Dusan Bogonovich's lab, looking at monoallelic expression, there's now evidence that if you have skewing towards the mutant allele, where just the allele that carries the mutation is expressed, then that can influence the phenotype. And then there's a number of cellular mechanisms that we should consider when thinking about, are somatic variants pathogenic potentially? So they could have a very dominant effect, and we'll talk about that very briefly with TLR8 in a moment, where you have a small number of cells affected, but they have a very strong response on the immune system. There could be some type of a selective survival advantage where the genetic variant that the cell has leads to either proliferation or survival of a particular cell type that mediates disease in the patient. And there's actually been a few cases that are relatively rare where loss of function in a subset of cells is enough to tip the patient over towards having disease. For example, this has been described as loss of function mosaic variants in chronic granulomatous disease, for example, or in STAT3 loss of function. Again, very rare. And I just want to give an example of a dominant effect of mosaicism in TLR8 gain of function, because I think that sometimes when we think about genetic mosaicism, and much of the work that's been done in the field has come from cancer, which has really given us a lot of tools to examine this. But also, there's a presumption there that mosaic variants are going to lead to some type of survival advantage or outgrowth of a cell type that has the variant, and that's not always true. And so TLR8 gain of function is associated with mosaic or germline variants in TLR8 that lead to increased signaling. Mosaic variants we've described in patients between 7 and 26% of the cells. These patients have lymphoproliferation, they have neutropenia, low B cell numbers, and ultimately progress to bone marrow failure. This is a hematopoietic disease. Hematopoietic stem cell transplantation is curative, although it's unfortunately has a relatively high mortality rate prior to transplantation. And it is also a dominant disease. So this is X-linked. TLR8 is included on the X chromosome. However, we've identified 2 females now, one germline, who we published in our most recent publication on TLR8 gain of function, and another patient that we've recently identified who has genetic mosaicism in TLR8. These are the variants that we see in TLR8 gain of function, just focusing on the red and the black. This is the phenotype of what we term infiltrate, or this syndrome of lymphoproliferation and bone marrow failure. You can see that they tend to cluster around this Z loop of TLR8. This is the endosomal portion of TLR8, which is a pattern recognition receptor that recognizes single-stranded RNA in the endosome. It functions as a homodimer. Most of the patients that we see have somatic mosaicism, shown here in red, in a couple of germline patients. And I really just want to bring this disease up because it has quite a dominant effect on the immune system. So we have not observed any selective survival advantage of an immune cell having TLR8. The transcript of TLR8 is limited to monocytes and other myeloid cells. Even in the case of patients who have these variants, we don't see expression of the transcript in lymphocytes. And if we look by ddPCR in different cell types from patients, just shown here on the top here, focusing in, we can see TLR8 DNA in fibroblasts, as well as in whole blood, saliva, NK cells, T cells, and monocytes. So this is likely an early mutational event. And that is shown here when we look at the variant allele fraction. So basically the number of reads in whole blood. These are all males, all XY males. And so this correlates to the percentage of cells as well that have the variant, and the age at disease of onset. And in stars here are variants where we've identified that they are mosaic, and the variants are present both in blood and immune cells and in fibroblasts, meaning this is probably a very early event during embryogenesis. They have had this for quite a while, and we don't see a correlation between the variant allele fraction necessarily in disease onset. So despite only a small portion of monocytes and myeloid cells expressing variant TLR8, there's a very dominant effect on the immune system. We see changes in T cells and B cell compartments. We see defects in memory T cells. We see expansion of CD8 TEMRAs, and we see very high levels of cytokines in the peripheral blood, indicative of both myeloid cell activation, such as high levels of IL-18, and T cell activation, such as high levels of interferon gamma and soluble IL-2. So TLR8 is one of about 20 or so known mosaic genetic errors of immunity. They're listed here. Not going to go over all of these, but I do want to emphasize that for the most part, most diseases lead to immune dysregulation of some sort because they're having some dominant effect on the immune system. Most disorders are gain of function or loss of function of a regulatory function. For example, loss of function of FAS, loss of that regulatory function. And mosaicism, there's a very wide range from anywhere from 1 to 80%, depending on the compartment that you're looking at and the disease that you're looking at. So just to summarize, genetic mosaicism can lead to monogenic diseases of the immune system or genetic errors of immunity. I think it's important to think about as physicians and scientists that there could be multiple different mechanisms by which a mosaic variant can actually lead to disease. It does not require that variant provides any selective survival advantage to an immune cell. So this is different from thinking about a malignancy. And identifying these variants really leads to changes in patient care. So just another tip there, always sequence the tissue where you might expect to find a variant. And thank members of my lab and look forward to the rest of the webinar.
Wonderful presentation, Megan. Thank you so much. I'm looking forward to the questions and discussions. Please put your questions, if you have them, in the Q&A session, and then we can discuss them after all four talks. It's now my pleasure to introduce Laura Batlle to you. Laura studied human biology in Barcelona before she moved on to Uppsala in Sweden to pursue her master degree. She specialized there in genetics. Back in Barcelona, she completed her PhD, where she focused on novel genetic mechanisms in primary immunodeficiencies, in close collaboration with actually the Radboud University in the Netherlands. She did her postdoctoral training at the Vall Hospital in Barcelona in Spain, and her expertise is not only in immunology but also in genetics and bioinformatics. Moreover, she is the ESID Junior country representative for Spain. So Laura, thank you very much for accepting our invitation to speak. We're looking forward to your presentation.
Thank you, Bodo, for this nice introduction, and thank you for having me in this webinar. It's really an honor. I think you can see my screen now.
Yes. Perfect.
Great. So yeah, today I'm going to talk about genomic technologies for somatic variant detection, and Megan has already gone over some of the main concepts, so that could be very helpful. And first of all, a concept that I think that needs to be very clear for the rest of my talk is variant allele frequency. Variant allele frequency is basically the percentage of reads that we have supporting our alternative variant. When we look at germline variant, either heterozygous or homozygous variants are expected, so 50% or 100% variant allele frequency. But when we are looking to somatic variants, we have a whole range of different percentages of reads that can be supporting the alternative allele. And what it makes it a bit difficult to detect using genomic technologies is that these ranges can really vary across different genes. So here, again, just to exemplify this concept, an IGB real case view where we can see how variant allele frequency is calculated. And as Dr. Cooper already mentioned, there's not a threshold where variant allele frequency is considered pathogenic. And I made this figure using actually Dr. Cooper's lab data, which is an overview of the different effects that has been described and the variant allele frequency that has been seen in these patients. And you can see that there's a lot of different variants and different frequencies that can occur. So it's important to notice that these variants are driving disease in some inborn. Actually, I really like the concept genetic errors of immunity better than inborn, especially in this context and in this webinar. So it's important to consider them when looking for genetic diagnosis. And also, it may really explain some unsolved cases that, given the current diagnosis rate using genomic technologies, it's really worth to look at this kind of variant. And as just shown with this beautiful example, it can also open the door to guided treatment and a better management of the patient. So here we have an outline of a genetic study, and it can be applied to any disorder. Actually, everything starts with the patient. We select a sample, perform sequencing, and then we do a bit of bioinformatic processing to just align the reads to the reference genome, call the variants, filter them, and hopefully select a candidate variant that we will then validate and report. When studying somatic variants, this is the standard pipeline that we follow, but we have to consider some things that are a bit different from germline variants. For instance, we have to be careful about which patients we do select and the probability of finding a somatic variant there. What are the tissue that we are studying? How are we processing the sample? How are we sequencing it and at which depth? And regarding the variant calling, we'll see that there are specific strategies that can help us, similar for filtering and then for validation. Regarding two reports, it's also important to specifically state in the report that we have a somatic variant which will have implications in genetic counseling. In order to detect somatic variants, this is a bit of a spoiler because the title is already there, but we can use different technologies. People do not think about Sanger sequencing when discovering somatic variants. It's always a bit of a debate if a Sanger sequencing is really outdated or still in use. I think that we live in different countries, we work with different technologies, and we have to work with what we have, and we have to work hard to find a diagnosis of our patients. So I'm here a bit to demonstrate that even with basic technology, you can still look for this kind of mosaicism. So here we have different Sanger sequencing from different patients that we've been analyzing in our lab, and we can see that this technology can really detect a broad range of variant allele frequencies, from a very small fraction of cells having the variant. In this case, we were lucky enough that it was an insertion, so we can see the different peaks, to an increasing frequency. This is useful in some contexts, especially when you already have the sequencing data and you can revisit it in order to know if you can reconsider some of the diagnosis, and it's quick and easy, but obviously it's not ideal. It requires some expertise because here the sequences are quite clean, but usually we have some noise that may be a bit difficult to sort out from our real calls. And also it's not quantitative. So here we see some numbers, and this has been validated by NGS. But if not, we cannot really say which is the variant allele frequency from these chromatograms. But it's good as a starting point, and I think that it's a tool that some of us already have. So it's good to revisit these results. We move now to NGS, and here we have the whole range of genome, exomes, and panels. In order to detect somatic variants, we'll see later that the best strategy that we can do is to sequence deeper. So target sequencing is usually the way to go because we have more reads supporting the genes that we have picked for our panel. It will also depend on how deep are we sequencing this panel, how big, how many genes are we including, but usually this will be greater than whole genome, that in terms of cost will be extremely expensive to sequence at the depths. And then for exome, we have regular exomes, but now deep and ultra deep whole exome sequencing is also available, so it may be worth in the cases where we do not really have candidate genes. Again, here just to illustrate a bit the importance of coverage, we have here in green a variant. If we have low coverage, we find only one read carrying the variant, and that could be a real variant, could be noise, could be a sequencing artifact. But if we have more reads supporting the variants, this will be more likely a true variant. Here's a real example of this very same case, and we can see that we have only 4 reads supporting that variant. So just some tips in order to, when looking at IGV, what we can look for to know if this is a real variant or not. I will try to look at the mapping quality of this variant, the base quality of the specific position, the balanced strand representation, and also if the variant is present in different positions in different reads, which can indicate that the variant is real and it's not an artifact. Regarding how we process the data, obviously we do not do variant calling by looking at IGV and going through variants. We use colors, and usually at the genetics labs, we use a germline variant caller that will be picking up the variants that are 50% or 100% viral frequency, so heterozygous or homozygous variants. This is useful. It's the main way to get to the diagnosis in many patients. But for some patients with somatic variants can also still be a tool that we have and that may be used. So here we see a part of the exon 9 of the FAS gene in an ALPS patient, and we see that we have some calls. These calls are at the very, very low variant allele frequency. I don't know if you can see it here, but it's below 1%. And in all these calls that we can see, we see one that is a bit higher up and the other ones are very, very low. But we have not distinguished exactly what is a true variant, what is not. We have other strategies for calling these variants, for instance, using a somatic variant calling software. We have different options such as Mutate2 or BarScan2. And in this case, we start from the very same input file, a BAM file in this case. And here you see in red that now the number of calls has really multiplied. When we look for somatic variants, the parameters that we're using, I'm not going to enter into much bioinformatic detail, but basically we will pick all the variants regardless of their variant allele frequencies. So we'll have even more noise. We can see that we still have this call up there that seems real, especially because it's replicating in both colors. That's a very good sign as well, but we still have a lot of noise. So here again, some tips for filtering this out. Of course, look at the quality of these calls, look at the number of reads, alternative reads, total reads. Evaluate the variant effect. If we're looking for pathogenic variants, we can use many predictions, filter for frequency, and different things. As if it was a germline variant, we can also apply ACMG guidelines to classify the variants, although there are some specifications that we need to take into account. And if you have several patients, what you can also build that you know that appear to have somatic variants that may be artifacts because are repeated in all your patients if they do not have all the same disorder, which is frankly very unlikely. Also, a strategy that we really like is to use pair samples. Following up with our ALPS patient, we know that somatic variants in ALPS are located at double negative T cells, and these cells are a very small subset of cells in healthy individuals, but are expanded in ALPS patients. These cells are inside the CD3 plus marker, and we will find these cells even more diluted in whole blood. So the strategy here is to sequence in parallel 2 or more tissues in order to know if the frequency of this variant is increasing as we would expect. So here we see first a plot of the variants with a somatic variant color. We've seen already that, and we've seen this peak up there, but we didn't know even after our filtering if this was pathogenic or not. Performing exactly the same experiment, but now using CD3 cells, we can see the very same peak, but in the frequency higher up, so more or less doubled. This will most likely indicate a true somatic variant, because what we are seeing here is that the noise do not increase in frequency when using CD3 cells because it's the same variant and the same random noise. But when we have a real variant, it really shows at an increased frequency. We can enrich our sample using different methods, and this will also depend on what you have available of your lab. Centrifugation methods are really quick and quite easy. Magnetic beads are also super useful through negative and positive selection. And of course, the gold standard will be sorting. We can obtain our population of interest and then extract DNA and sequence. Just be mindful here of the volume that you will need for that, because if you sell your target population has only very few cells, you will need a lot of starting volume in order to have enough to extract this DNA and sequence. Okay, as I have already mentioned, a good way to improve our sequencing capacity to detect these somatic variants is to sequence deeper. We can do that in different manners. We can design targeted panels, as they have done beautifully in Dr. Cooper lab. There's also some commercial solutions for that. So they just pick a panel of genes where somatic variants have been described, or autosomal dominant genes where somatic variants are suspected, and sequence them at high depths. We can also perform amplicon sequencing. If we have a targeted region, we can design primers and amplify that. And this has been beautifully shown by Ana Mensah-Vilaró a few years ago. We can also perform molecular inversion proofs, which will be even better than amplicons because they have a backbone that will ensure that we don't have PCR duplicates. This has been done in autoinflammatory disorders, and we've also set up this technique for FAS in collaboration with Alexander Hoshen at Batlle University. Finally, a good way to validate our calls, and we already seen this in the last talk, is digital droplet PCR. I'm not going to enter into details, but this is a great way to validate our variants and to specifically quantify which is the variant allele frequency. Just be mindful that for doing that, we need to know specifically the variant that we're looking for, because we have to design the primers. So this may not be very useful for discovery unless there's a very strong hotspot in the gene that we're studying. Okay, and finally, I did a bit of a recap of important considerations that I think are important when setting up strategies to detect somatic variants. First, I've been saying somatic variants for a while now, but really the term mosaicism would be a bit more correct here, because we have these mosaicisms that can be inherited in a gonosomal or gonadal mode, and then the so-called true somatic variants, where the variants occurs in the embryo or postzygotic. This will affect drastically the amount of cells that are affected in the individual and also the recurrence risk. So it's quite important to do not assume that the variant is de novo, to test the parents and test the rest of the family if they are available. Also, just a big flashback to embryology class, and be mindful that different tissues in adult bodies all depend on the embryonary origin. So during gastrulation, we have these 3 tissues, and all the tissues that we have nowadays are derived from that. When testing somatic variants that can be present in different tissues, it's important to know the origin of each of them, either to validate if the variant is present or not there, or to see at which time point the variant appeared. So here I have an example from a paper that we wrote a while ago, where we see in each line a variant, and some variants really do have a similar frequency across tissues, meaning that they appeared early on development. But for instance, in green, you can see that some variants have really different variant allele frequency in different tissues. So it's worth checking and making sure that you are looking for the variant in the tissue where it's represented. And finally, a quick word about treatment. Working with ALPS patients, we have realized that some of them really start treatment early on, and it's a bit tricky for genetic testing. People usually assume that DNA is a very fixed thing, but as Dr. Cooper already said, this is not true for mosaicism. So in this case, if we start immunosuppressive treatment, we'll have a decrease in double negative T cells and also a decrease in the variant allele frequency. So my advice here will be to test pre-treatment samples or DNA, or at least store them in case you will have these studies available later on. And this is also true for many functional validations. So with that, I reach to my conclusion slide. And some points that I think that may be interesting, it's that accurate diagnosis of somatic variants requires detailed knowledge of disease biology and the relevant affected tissue or cell lineage. High-depth NGS combined with dedicated somatic variant calling algorithms is an effective strategy for identifying low-frequency variants. Targeted sequencing approaches are essential to achieve sufficient sensitivity while managing data complexity and cost. Enrichment of the relevant tissue or specific cell populations improves variant detection, enables more accurate variant allele frequency estimation, and supports validation. And finally, familial studies remain important for appropriate interpretation and genetic counseling. And with that, I just want to thank my group, especially to Roger Colobran, who has been key in all these studies, and also to all of you for your attention. Happy to take questions. Thank you.
Thank you, Laura, for this stellar talk. We'll keep the questions for after the 4 presentations have been given. So wonderful presentation. Thank you very much again. And I'm sure we'll come back for questions. But now it's my pleasure to introduce Sinisa Savic. Sinisa did his physician training at Newcastle University. And after that, he did his PhD at the University of Leeds. And then in Leeds, he became professor of clinical immunology in 2023. Sinisa cares for patients with primary immunodeficiencies, but also for patients with systemic autoinflammatory disorders and allergies. In his research lab, he performs studies on genetics of these rare inborn errors of immunity and also on autoinflammatory diseases. And he's contributed to several discoveries in the field. So, Sinisa, we are very much looking forward to your presentation on VEXAS syndrome.
Thank you, Bodo. Thank you for the invitation to speak. Let me just share my slide. There we are. Okay.
Coming.
Yep.
Perfect. Now
Hopefully. Okay. So I'll try and get through this in 15 minutes. And if I'm running over, please let me know and I'll stop. So it's a pleasure to speak about VEXAS syndrome. It's a remarkable condition which has really shaped, in particular, the recent few years of adult practice in immunology and really changed the ways how we think about some of the not just rare disorders, but also some of the more common rheumatological conditions. So I thought the best way to introduce this condition is really just to talk through one of the cases, which is one of the original patients that was eventually diagnosed with this condition, because it really illustrates quite nicely the presentation, the clinical course, the complication, and the difficulties we still have in managing these patients. So this gentleman who has sadly now passed away, he presented originally at the age of 52 with a fairly non-specific set of symptoms that included flu-like illness, episodic fevers, night sweats. He developed this very particular rash, but again, some arthralgia, myalgia, and had this fleeting shortness of breath associated with lung infiltrates. And as many of these cases are, he was extensively investigated, and all the typical autoimmune and infective causes for his symptoms were excluded. He was found to have mild macrocytic anemia. He had this elevated CRP, which had continued to be the feature throughout his illness. And the PET-CT was really not that helpful. It showed mild bilateral lymphadenopathy. And because of that, and this rash, he was given this provisional diagnosis of systemic sarcoid and treated with corticosteroids, to which he made a remarkable recovery. And then every time the corticosteroids were reduced and some form of conventional DMARD was introduced, he would relapse and really remain dependent on corticosteroids. Then he came to my clinic and I saw him and I said, look, seems to me that you have one of these, autoinflammatory disorders, the cause of which we still don't understand fully, but nevertheless, it's certainly possible to treat you empirically. And one great treatment that I often find highly effective in these cases is using anakinra. And 10 days after initiating anakinra, he had this really awful injection site reaction, which is I hadn't seen in any other conditions other than VEXAS syndrome. It seems to be very typical for this. And he had a very stormy course where he basically had multiple severe infections, which landed him in hospital, gradual decline in bone marrow function to the point where he then required blood transfusions. And all that time he's been treated with a combination of tocilizumab, JAK inhibitors, and other anti-inflammatory targeted therapies in attempt to control his symptoms. And all of that, to some degrees, worked, but not really was in any way curative or caused major disease remission. And then he was found to have this very particular mutation in the gene UBA1, which is at the position methionine 41, which was consistent with diagnosis of VEXAS syndrome. By that point, he was quite unwell and really didn't respond well to any of the targeted therapies that we had treated him with. He then was considered for bone marrow transplant and actually underwent the bone marrow transplant, but sadly died early in the post-transplant period due to complications. So this, in a nutshell, really illustrates many of the important features of the condition, the difficulty in managing these patients, because on one hand, they have heightened inflammatory response, by the same time, as I will come to show you, they have degree of immune deficiency, and actually is a secondary immunodeficiency condition that we are trying to manage alongside. So the VEXAS stands really for V vacuos, which is typical finding on progenitor cells within the bone marrow. It's not the only finding, it's not pathogenic for VEXAS, it's found in other conditions, but in VEXAS syndrome there seems to be more of these vacuoles and they're slightly larger. E stands for E1 enzyme, which is the function of the UBY gene that is mutated. X-linked because the gene was found is on the X chromosome. Autoinflammatory is for A because of the symptoms are in keeping with autoinflammatory condition, and S as somatic because the mutation is not inherited but it's acquired later on within the bone marrow. So this is really the kind of summary slide of pathogenesis of VEXAS syndrome. As you can see here, for reasons which are not entirely clear, there is a mutation, and as was shown earlier on by Megan, obviously as you age, there is a more chance of this happening. And as you will see, the vast majority of patients with VEXAS syndrome are elderly, and in particular male, because it's an X chromosome related gene. You have the mutation at this very particular point, methionine 41, and that's essential for the disease manifestation because this is a translation codon for cytoplasmic form of the UBA1 gene. So the UBA1 is translated in two isoforms, nuclear and cytoplasmic. If you have a mutation at methionine 41, you lose the initiation signal. In essence, you lose the expression of UBA1B, and you have expression of something shorter, UBA1C, which is enzymatically inactive. But UBA1 enzyme is critical for the whole ubiquitination pathway. It's there at the beginning. It has E1 ligase activity, and it's essential for 95% of all the ubiquitination, initiation of all the ubiquitination that happens in the cell. Consequently, there are multiple cellular functions, including regulation of inflammatory pathways that are affected as a result of this. Separate to the inflammatory complications, patients develop bone marrow failure, which is not entirely clear. There are many potential explanations, and I'm not going to necessarily go into details about it at this point. And it's been already alluded, these mutations can happen anywhere within the hematopoietic stem cell system, but in VEXAS, actually, these mutations do happen at the level of hematopoietic stem cell progenitors, and they can be found in all the lineages. But interestingly, it is only the myeloid cells that can survive with the mutation in the periphery. So your T and B cells that are found in periphery of these patients carry wild type. They don't. The lymphoid progenitors that carry the mutated form of the protein cannot survive. But interestingly, neutrophils and monocytes can, and these are the cells which cause most of the inflammatory pathology in periphery. And this is illustrated on the next slide. Sorry, I'm just not sure what's happened here. Right. Seen on this slide here, that basically you can very simply, when you're just looking at the VEXAS neutrophil, it looks completely disordered, disorganized. And when you study the neutrophil in more detail, you find that there are many inflammatory pathways that are activated, which really explains why it's difficult to treat this condition with a targeted therapy. On the other hand, here, if you were to biopsy the involved skin of the VEXAS patient, you'll find that the vast majority of cells that are infiltrating in these lesions are mutated cells. And this is a similar process that happens in other sites and tissues. A lot of the inflammatory complications are mediated by infiltration from the neutrophils. The immune abnormalities that are seen in VEXAS syndrome are many, and these two papers have nicely described in much more detail, which I'm not necessarily going to go into, and you can go back and read through this. But in essence, you have on the innate immune side, exhaustion of the monocytes, increased activation of inflammasome, increased production of IL-1 beta and IL-18, and overall enhanced NF-kappa B signaling. But there is a broader immune dysfunction, and due to this kind of inflammatory soup, there's a lot of skewed and exhausted cells, even of the adaptive immune system, and that all contributes to the phenotype that we see in the condition. So other than the mutation of the methionine 41 position, there are other rare variants now being picked up in the UBA1, but their role is somewhat less clear. And we and others have identified a number of rare variants. Again, the allele frequency of these variants vary anywhere from few percentages to up to 70, 80%. But we find that most of these other ones, which are not affecting methionine 41 position, are more associated with myeloid type conditions like MDS and AML. And actually, some of the earlier VEXAS patients who have been described with these non-methionine 41 variants, they have a slightly different non-inflammatory, more kind of hematological clinical picture. So how frequent is VEXAS? And this was a question which was asked pretty much straight away after the condition was discovered. And David Back, who led the discovery of VEXAS, did another study shortly afterwards in the States using this particular Ganzigen code community cohort of individuals who seek healthcare for various reasons. And in that cohort, he found that there was up to 1 in 5,000 potential cases of VEXAS in men over the age of 50. There were also some female cases and much higher, lower frequency. But subsequent study, again in the US, led this time from a different center and a different group, didn't really replicate this in the same way. They did find a lot of methionine 41 mutations, but largely in women. And these women actually, to this day, as far as we know, don't really have any clinical features, neither VEXAS or any other common conditions, and actually found few these pathological mutations at very low allele frequency in otherwise healthy men. So the picture wasn't entirely clear in terms of frequency. We did another epidemiological study in the UK. This is where we were using basically UK Biobank and also actually taking the advantage of the diagnostic labs, which by that point were routinely sequencing and screening for the UBA1. And essentially, to cut a long story short, we think, well, from our findings, we estimated about 171 new cases of VEXAS syndrome in men over 50 who have been investigated for unexplained anemia or myeloid disorder. So this is in a population of around 50 million people. You might see about 170 new cases each year. The other point to make is that VEXAS is not, is now being described pretty much in all continents, in all ethnic backgrounds and so on. So it's not a disease reserved for Western Europe or North America. It's found everywhere, and it's unsurprising given that there's no reason why similar pathology cannot happen anywhere else. So this is just to remind us of that. And also, you can actually see female cases of VEXAS syndrome. Now, up to now, in the literature, the vast majority of the cases that have been described are in situations where you have 45X0 or X monosomy situation, which in essence give you the similar background to, in that respect, as being male from the point of view of acquiring just one additional mutation and causing the pathology. As far as we know, UBA1 is not subject to random X inactivation. So in essence, women with 2X chromosomes can compensate. But we did find one case where there was a female patient with 2X chromosomes in whom we did find a VAF 33% of one of these classical methionine 41 mutation with clinical features that were consistent with VEXAS. So if you have somebody who's female with features that may be suggestive, I would still urge for further testing. So what the clinical features and the disease course are now beginning to be better understood. This is a systematic review that was done by Adam from my research lab, and he basically looked at over 700 cases that were published up to that point. And as you can see, this is really what VEXAS looks like for most cases. Predominantly, they're all men, usually over age of 60. skin manifestations are very frequent, but we also mustn't forget the hematological part of the disease, which is that about 30% of these cases of VEXAS syndrome have frank myelodysplastic syndrome and actually have other mutations in addition to the UBA1 that are seen in MDS. They also all pretty much have degree of macrocytic anemia at the start, but over time develop other cytopenias and so on. There is increased tendency to develop venothrombotic events, and the DVTs and PE are frequently described. So this is sort of broad overview of the main kind of features, but there are other manifestations such as some patients presenting with HLH, which is again being described. And variable allele frequency was mentioned before, and as I said, when it was first described, the VEXAS syndrome was seen in patients who only had like 10% VAF up to the ones who had 80%, and seemingly at the time when we had limited numbers, there didn't seem to be much correlation between the size of the clone and the severity of the disease. But there is an emerging evidence to suggest that actually as the clone size increases, the disease severity becomes worse, and also there is a more chance of developing bone marrow failure. You can also use the clone size to look at the effectiveness of some of the therapies that target the clone, including the azacitidine, which I'm going to mention later on, and obviously the bone marrow transplant. I mentioned this already, but it's now quite clear that without any treatment over time, many of these patients will develop increasing bone marrow failure, become transfusion dependent, and not just one type of cytopenia, but multiple cytopenias. Another important point to make about VEXAS syndrome, which I alluded to at the beginning, is this high propensity towards the infections. And as I said, this is immune deficiency, VEXAS, and it's due to several reasons why these patients are immunodeficient. Firstly, they're cytopenic. So even though the myeloid cells do survive the mutation, often they are neutropenic, they're almost always lymphopenic. And the cells, the neutrophils which are found in periphery, this is the work that we are currently doing. We've shown that these cells are not efficient at killing bacteria. They have reduced reactive oxygen species production and ability to phagocytose. And on top of all of that, we give them all sorts of immunosuppressive treatments. So it's not surprising that overall the effect of all of this is that these patients have frequent infections. Actually, quite often is the reason for their reduced survival. And in addition to normal infections, many of them have been presenting with opportunistic infections, including PCP and mycobacterial infections. So briefly on management, I think the important point is who you should screen for VEXAS, and there isn't really one specific list that you can follow. There are many people working on machine learning and other ways of trying to come up with the best predictors who you should screen for VEXAS, but I find this relatively short list and this kind of diagram useful. It's still man over age of 50 or 60 that you're looking to screen, and it's really these cases who present with VA systemic inflammation for which you're struggling really to find a diagnosis, and in whom you might find a degree of anemia or some kind of cytopenia that should really trigger screening for VEXAS syndrome. And when you think about treating these patients, I think the important point, and this is something that's coming up, we have recently collected our experience of managing VEXAS patients through the national MDT that we have set up in the UK. And it's highly, highly helpful to do this because these patients can present to dermatologists, rheumatologists, hematologists, immunologists, and they will have various body system involvement, and you need collective, if you like, experience in terms of, first of all, recognition, screening, testing, and then as well as deciding on the best management, because it is not only the treatment of the inflammatory complications or trying to deal with the clone itself, but it's also the supportive therapy that is important to provide. And a point to make here is that, unfortunately, we still don't have an agreed protocol, in terms of when and what antibiotic prophylaxis should you start, which vaccination you should consider, at which point maybe you should consider anticoagulation and so on. And this is something which is currently emerging. But having an MDT where you can discuss these patients and provide support for various colleagues that there are centers where they may have only 1 or 2 cases, and there are centers where there's certainly much more better experience in managing this. And you can look at this in more detail. This is something that we published not that long ago. This is based on international consensus. It is a guidance, not a guideline, because it's based on an opinion, not necessarily a huge amount of prospective evidence. But in essence, it gives you kind of a template on which you can develop your algorithm for treating individual cases. Again, an important point to make here is that some patients, particularly elderly with comorbidities, can be managed with corticosteroids alone. And actually, up to 10-15 milligrams of prednisolone may be just enough to manage most of the inflammatory symptoms and actually keep them well enough and without necessarily needing any other treatment. But for younger patients who have maybe more severe disease, early consideration of bone marrow transplant should be there. There are other therapies which are either targeting inflammation or this treatment, which is usually used by the hematologist for treatment of MDS, which is azacitidine, which has been shown to have significant effectiveness in subpopulation of these patients. So I don't know how much more time I have, because my title was VEXAS plus other similar things. I have 5 minutes. I can talk about 3 minutes, right? Okay. Just quickly, beyond VEXAS. I mentioned myelodysplastic syndrome, and there are a number of other mutations that you can acquire within the bone marrow that can give you inflammation. And actually, it's been recognized long before VEXAS was recognized as a disease entity. It was recognized that some patients with myelodysplastic syndromes do have frequent autoimmune and autoinflammatory complications. And we did this study some time ago looking at patients with low risk MDS to see really, is there any evidence of inflammatory pathway activation? And in a short summary, yes, there is. We were able to, looking at the ASC NLRP3 specs in peripheral blood, show there is inflammasome activation, highly activated in these patients at a level very similar to what you see in CAPS. Multiple inflammatory cytokines are elevated, and actually some of these cytokines, IL-6 and IL-1 beta, may predict need for bone marrow for transfusion. And just to finish off, this is what the picture is now beginning to evolve further. These are the 2 cases that we are currently working on of patients who presented with VEXAS-like condition. But in their situation, they were UBA1 negative, but had the mutation in these 2 other genes called IDH1 and SRSF2. And these are genes that these cases are now being increasingly recognized in other centers. So you can present with very much VEXAS-like condition, but not necessarily due to UBA1 mutations. Actually, these 2 genetic mutations are found in patients with MDS, and quite often the patients will then progress to acute myeloid leukemia. And similarly, like in VEXAS, when you look at, just one snippet of the work we've done, these mutations are largely found in myeloid lineage. And again, supporting this idea that some of these, again, of the mutations, if they segregate to myeloid lineage, may really drive this kind of autoinflammatory type picture. To summarize, so VEXAS syndrome there's this now new terminology, hematoinflammatory conditions, really to highlight this overlap between inflammation and bone marrow dysfunction. The canonical mutation affecting methionine 41 is associated with typical VEXAS. These other mutations in UBA1 do give slightly different clinical phenotype, which is more of the hematological-based conditions. Immune basis of VEXAS is complex, as I explained, and there is a significant degree of immune deficiency with these patients. And the best treatment options and how to manage these patients are still emerging. And these are some of my lab members and people I collaborate. And thank you so much for listening.
Thank you so much for your wonderful presentation, Sinisa. This was a wonderful introduction, not only into the clinics, but also in the genetics. And thank you also for the management of these patients, as many of us struggle, to give them the best treatment they deserve. So wonderful presentation. Thanks again. And we'll finish off with a report from Roger Colobran. A surprising finding he did in his group. Roger has a degree in biology and a PhD in immunology. He works at the Vall Hospital in Barcelona, and he works in the field of immunogenetics. In his immunology service, he provides care for clinical and molecular genetics for patients with inborn errors of immunity and primary immunodeficiencies. And his clinical work he combines with the research in genetic mechanisms. And what he came across in recent months, he will report on. The floor is yours.
Okay, thank you very much, Bodo. It's a real pleasure to be here in this such an interesting session. I will share my screen. Okay, is it working?
Perfect.
Okay, so far we have seen how somatic mutations can cause inborn error of immunity or genetic errors of immunity. And now we will see, let's say, the other side of the coin with the mechanism of somatic reversion. So the definition of somatic reversion is pretty simple. It's a spontaneous post-zygotic genetic correction of a pathogenic mutation, typically inherited, resulting in a mosaic individual with a mix of revertant and functional cells. It is important to consider that this is a cell-autonomous event, so somatic reversion occurs in a single cell, and then for this somatic reversion to become apparent, we need to at least two requirements. One is that the revertant cell have to acquire an advantage to selectively grow and produce a cell lineage of these revertant cells. And for this to happen, the cell must become to a regenerating organ, typically blood, which is the tissue in which more somatic reversion have been described, but other tissues like skin and few others have also some regenerating capacity. So regarding nomenclature, you can see here that somatic reversion, the strict definition is the reversion of the mutated allele to the wild type. This phenomenon is also known as a back mutation, but there is another concept which is somatic genetic rescue, which is more wide and include any type of mutation that affect the gene itself and occasionally other genes that indirectly can restore the affected pathway. So these are more or less equivalent concepts, and I will use both during the presentation. When to suspect somatic reversion? Well, it's difficult to say, but typically when your patient experiences a quite milder phenotype than the expected. Honestly, probably in most of the occasions, this milder phenotype will not be explained by a somatic reversion, but by other factors. But still, somatic reversion is a factor that you can consider. Somatic reversion is a random process, but in some diseases in which the underlying mechanism results in genomic instability or favors a high mutation rate, obviously this also favors the occurrence of this somatic reversion. Here in this figure, this is from a beautiful review, you can see different mechanisms that have been reported for somatic genetic rescue. These mechanisms are point mutations that can occur in the exact position on the mutation or in the second site positions, or loss of heterozygosity mechanisms or some structural variants. Not all these mechanisms can be found in all the diseases because depending on the inheritance mechanism, some mechanisms can apply or not. I will give you just a few examples. For example, imagine an autosomal dominant disease that acts by haploinsufficiency. We have here this loss of function mutation in one allele. So in the other allele, a mutation in the promoter can be activating and somewhat compensate the haploinsufficiency by producing more quantity of the wild-type allele. Obviously, this is not possible in an X-linked disease in a male because we don't have the wild-type allele, neither in an autosomal recessive disease. So there are different mechanisms, and depending on the inheritance, can be produced or not. And what about somatic reversion in inborn nerve of immunity? In this table, you can see some of the diseases in which somatic reversion have been described. In fact, the first description of somatic reversion in human disease was in an ADA patient, and subsequently, other diseases have been reported. Interestingly, the probability of finding a somatic reversion is not the same in all these diseases. There are at least a couple of inborn nerves of immunity in which the somatic reversion is more prevalent. Briefly, for example, in DOCK8 deficiency, this is a very striking case because in the published cohorts, up to 40 or 50% of the patients have somatic reversion events. In these patients, as you can see here, you can find mutant cells that do not express DOCK8 and cells that express DOCK8 in a comparable levels like in a healthy control. So the repair mechanisms in these patients are different, but most of them rely on recombination events leading to gene conversion, or for example, in this example, intragenic single crossover, in which you have here two mutated alleles, and due to this crossover, then you have a wild-type allele and another allele with the two pathogenic variants. So why in DOCK8 deficiency there is so high prevalence of somatic reversions? Probably for different factors, but the main one is the location of DOCK8 gene in a recombination hotspot characterized by subtelomeric repetitive sequences. Curiously, these repetitive sequences are in most cases the responsible of the disease because most patients have these CMVs or structural variants, but the same mechanism favors the appearance of somatic reversion. The second example is the Wiskott-Aldrich disease. There are at least 40 cases published. Here is a summary of different cases published with different types of reversion. And this can occur in up to 10 or 11% of patients. And in this case, the mechanism is not well demonstrated, but probably the deficiency in GWAS protein causes somewhat genomic instability that might facilitate these somatic reversion events. And some repetitive regions of the GWAS gene can lead to this phenomena, which is called DNA polymerase slippage, that promote some indels that can restore the frame shift variants, restoring the reading frame and enable this somatic reversion. So after these general considerations, let me tell you a recent story that we experienced in our lab. This story is about a patient that we studied for many years. This patient was from a consanguineous family, and in 2012, she developed a severe episode of hemophagocytic lymphohistiocytosis due to an acute Epstein-Barr virus infection. At that time, we sequenced the 4 genes associated to familial HLH without any positive result. Due to the severity of the disease, the patient was transplanted from her HLA identical brother. The transplant was successful, let's say the procedure itself, but the patient was unable to control the virus during the next 7 years with multiple clinical complications due to this chronic active EBV disease. In year 2020, the patient experienced a spontaneous decrease in the viral load and remained clinically asymptomatic during the next years. So after this first genetic study in 2019, we put this patient in a research project, and by whole exome sequencing, we identified a pathogenic variant in this gene called tumor necrosis factor receptor superfamily member 9. It's a very long name, but it's more well known as CD137 or 4-1BB. 4-1BB is a positive co-stimulator for T cells. Maybe you know this molecule because it's been widely used for the CAR T cells, the chimeric antigen receptor T cells for cancer immunotherapy. In any case, it's a potent co-stimulator for the T cell response. The mutation was an stop gain mutation in the C-terminal domain of the protein that abrogates an important TRAF domain for the signaling. Few months ago of this finding, 3 consecutive papers described the CD137 deficiency as a novel inborn errors of immunity in patients with chronic EBV infection and EBV-induced lymphoproliferation. So the phenotype of our patient was highly concordant, and we considered that we found the disease-causing mutation. Then we wanted to do the familial testing. At that point, the family refused the genetic testing. It was a family with some cultural differences, and they refused to do this testing. But we were worried about the genotype of the brother who was the transplant donor. So at that time, it's year 2020, the patient had a mixed chimerism in B cells, in neutrophils, and almost a full donor chimerism in T cells. So we decided to test the status of the mutation in the blood of the patient, assuming that there will be a mixed population from the donor and the recipient. We did Sanger sequencing, and you can see here, this is the position of the mutation. This is a reverse sequence. This is the stop codon produced by the mutation. And in the position of the mutation, there is no trace of wild-type allele, indicating that the brother was probably homozygous for the pathogenic variant. We will see some details later. But if you carefully see at the next position, you see this little peak, which in some occasions can be confounded by background noise. But as Laura mentioned, if you have some expert eye, you can suspect that it's not just background noise. We repeated the PCR with different primers. The peak was there, and the peak was not present in control DNA. So we suspected that this could be a somatic revertant because this peak turns the stop codon into a missense variant that could eventually be less deleterious. So a lot of questions arose from this experiment. Let's see first the brother. Finally, the family agreed to test the brother, and the brother was indeed homozygous for the pathogenic mutation. We did the test in blood and in saliva, and you can see here that there is no little peak in the previous position. What about the brother? So the brother was homozygous carrier of the mutation. He was clinically asymptomatic, although when we did EBV copies in blood, the brother had slightly increased copies, but not clinically relevant. As a curiosity and relevant for this study, in one of the 3 papers that described the defect, they reported 4 consanguineous families, and in 3 of them, they reported 1 homozygous clinically affected patient and 1 homozygous clinically asymptomatic sibling, so in 3 families. So it seems this is a striking case of incomplete penetrance, which is not frequent in these autosomal recessive disorders. If we go back to the revertant, we tested the saliva of the patient, and the peak was not present. And we tested the DNA that we use for the whole exome sequencing, that was DNA previous to the hematopoietic stem cell transplant, and the peak was not there. So this confirmed that this is a somatic event that occurred post-transplant and only in the hematopoietic compartment. Then we isolated TMB cells, and you can see here that the somatic reversion is only present in T cells and is completely absent in B cells. You can see, as Laura Batlle mentioned, that the peak of the blood is enriched in the target population. And in fact, it's not only enriched, but you can see here another little peak in exactly the mutation position that could represent a second revertant, that in this case, it would be a true revertant, so a back mutation turning the stop codon into the wild-type sequence. So it seems that we have 2 revertants, and we quantified, in this case, by deep amplicon NGS sequencing, the frequency. You can see here, the revertant 1 has 5% frequency in the blood and is enriched in T cells up to 40%. And the revertant 2, 3% in blood and 8% in T cells, and no presence in B cells. But the question is, are these really 2 independent revertants, or maybe the revertant 2 arose in the revertant 1 cells? To solve that, we examined the IgV reads, and we confirmed that indeed these are 2 independent revertants. You can see here, most of the reads carry the mutation. Some reads carry the mutation and the second site reversion, leading to a missense variant. and few reads have this back mutation transforming the stop codon into the wild type sequence. So the patient has in her blood most of the cells are mutant cells, but she has two populations of revertants. The other question was when these revertants are formed. We know that at some point after transplantation, but at which point? This is a very difficult question to address, obviously, but we could partially address recovering the DNA from the chimerism test. So we contacted with the Blood and Tissue Bank of Catalonia, and we were able to recover different DNA samples of purified B cells, T cells, B cells, and granulocytes from different chimerism tests. Here you can see the chimerism, and we were able to trace back the mutation until 2018. In 2018, the revertants were already there, but the frequency increased, you can see here, until year 2020, and then started to decrease again. Again, there is no trace of the revertants in B cells and granulocytes. And finally, so the revertant is in T cells, but in which T cells? We purified CD4 and CD8 T cells, and this is beautiful in my opinion, because you can see that the revertant is only present in CD8 T cells. And this makes sense because CD8 T cells are the cells that are crucial for fighting the Epstein-Barr virus. So we finally did one experiment of single-cell RNA-seq in CD8 positive cells of the patient. So we take 5,000 CD8 positive cells from the patient's blood. We did single-cell RNA-seq. You can see here the distribution of CD8 T cells according to the UMAP statistical analysis. These are the naive T cells. These are memory T cells, effector memory T cells, and these are effector memory RA cells. So our revertants are here, are two different populations, revertant 2 and revertant 1, but both populations belong to these TEMRA CD8 T cells, which is again concordant because these populations typically are expanded in viral infections, especially in chronic viral infections. Then we examined the TCR sequences of these CD8 T cells, and we classified the TCRs according their level of expansion. You can see here, this is again the graphic in which we see the revertants, and the TCRs can be hyperexpanded or less expanded. In naive T cells, the TCRs are not expanded, as expected, and both in revertant 1 and revertant 2 populations, the TCRs are hyperexpanded, leading to the hypothesis that probably these revertants were able to recognize and respond to the Epstein-Barr virus, creating these revertant populations with these hyperexpanded TCRs. And the last finding, which is again fascinating, is that we realized that all revertant 1 and revertant 2 cells were classified as male cells. So this indicates that the patient was transplanted from his homozygous brother, and then at some point post-transplant, some cells of the donor, but in the body of the patient, experienced this somatic reversion and expanded to create these two revertants. So this is my final slide just to illustrate the variant allele frequency of both revertants that we were able to trace during 7 years from 2018 to 2025. The maximum peak of expansion was in year 2020, correlating with the spontaneous, I say spontaneous, but you must understand that it's not spontaneous, it's because the revertants, this is spontaneous control of EBV viremia. Then some of the revertants decrease its frequency, and now the patient started to increase EBV viremia. We don't know what will happen in the future, but still at this point the revertants were able to at least more or less control this viral infection. So thank you very much to all the collaborators, especially Laura Batlle, that you have seen is an expert for somatic variant detection, and all the collaborators in different hospitals in which this patient has been treated. Also, Ferran Casals from the University of Barcelona, and Ana Esteban Joan Pedrosa from the SENAC, who help us in the single-cell RNA-seq experiments. And thank you very much to you for your attention.
Wonderful. Congratulations to this wonderful work, Roger. So this has been exceptional, and congratulations again also to the talk. Now, this concludes our 4 presentations, and we are now moving for the last 5 minutes to the questions. I find most of the questions have been already kindly answered by our speakers in the Q&A session. If you do not find your question anymore, I think you have to click on the middle panel to find your answers. But there are still a couple of open questions, and I think we start with Megan with a question towards Megan. What could be the mechanisms of low variant allele frequency in TLR8 causing the disease?
Hi, thanks. Yes, I do think it's a secreted factor, as Ashish was asking in that question. So I think that cytokines produced by myeloid cells, in particular monocytes and activated macrophages, are having an influence on the immune response. In particular, we see hyperexpansion of T cells related to that. And we can see if we derive macrophages from patients in vitro from their iPSCs that either have the variant or don't have the variant, we see very high levels of IL-6 and other pro-inflammatory cytokines from those macrophages.
Great, thanks. Megan, I think let's stick with you. I know that Laura can probably answer the same question. The next question is about in cases where we don't know or the causative cell types is not reported, how can we detect then the somatic mutation? Or what is the best approach to detect the somatic mutation?
So I think as Laura brought up, and we recently put out a report on this, I think probably deep sequencing is the best way if you don't know, and deep sequencing of the tissue of interest. So that's going to be blood for the most part. Although I think there's a lot of interest among our field and other fields in looking at other tissues, for example, looking at intestines for patients with inflammatory bowel disease, because that might be where you find the somatic variant. I think you're unlikely to find it by genome sequencing of a bulk cell population. Potentially by exome sequencing, there might be ways to detect it, but it's probably going to be variants with a VAF of 10% or higher, and you're going to miss the lower VAF variants of that. So I think for right now, practically deep sequencing. Of course, in 5 years, we might have deep exome or deep genome sequencing widely available. We'll have to see.
And if we stick with the whole blood, what would your suggested coverage to detect, let's say, a mutation with a 2% frequency?
I think you're going to need at least 1,000x of if you're kind of going for a discovery, if you don't have a hypothesis about what gene it might be. We, with our panel, do about 5,000x, and we can very reliably detect down to a VAF of 1%. And in some cases, a little bit lower than that, although you start to get significant noise under VAFs of 1%.
Thank you. Laura, can you contribute to the same question with your experience? What are current patients you have seen? How deep did you have to go?
Yeah, I completely agree with Megan, of course, that the deeper the better. But we also had some experience, for instance, in ALPS, as I've been talking about, where you have a very defined target exons. So we were studying only 3 exons, and then even if with shallow sequencing, let's say 200x like a regular panel, if you know what you're looking for, you can just pick the variants and then try to sort them from the noise. But this is, of course, having a lot of knowledge about the disorder and the candidate variants, which is not always available. In that cases, I would also go for deeper sequencing.
So if you order an exome with 1000x coverage, how expensive is that if you go for research?
I think it will also depend on the deal that you have with your provider, because this varies a lot. I think that now regular exomes are around 200 and cost around 3,000 euros. Sorry, 300 also, or 500. So I don't know. I will bet around 1K per exome, per deep exome. I don't know, Megan, maybe you have more specific data.
In the US, about 1,000 for a deep exome research basis.
Great, thank
Same
you.
here
Next
then.
question. Have you ever encountered mosaicism in a gene that has its own pseudogene? Oh, that's a difficult one. How can this affect variant calling and determining an accurate variant allele frequency? Laura, you go first.
Good luck to you. I think that's a very complex case. Pseudogenes are still a challenge using NGS. Some of the genes are not very well resolved now with long read sequencing. It seems that it's going to be a bit better, but for us, and I know that many other diagnostic labs still use Sanger sequencing for some of like NCF1 and these complicated genes. We have never worked with somatic variants in pseudogenes. Probably we missed them if they are there because we haven't really looked at that. But that's a very interesting case. Also, it reminds me of people looking for structural somatic variants, which is also a very interesting field.
Thank you.
May
Megan,
I
do you
add a
have any
comment?
experience with pseudogenes?
Just briefly, yeah, we're working on a case of IKBKG now, NEMO, that's somatic, and it's been challenging, but what we've done is amplified the region surrounding it and done long read sequencing. And then when you map back to the genome, if you mask the pseudogene, then you can map it back and get an accurate BAF. But again, you have to know what you're looking for.
Thank you. Next question is to Sinisa. What about SAMD9 ligand? That's also interesting to me because we always in our CVD patients find SAMD9 ligand variants. So the person who asks has two patients with different phenotypes and both had different VUSs. How can we evaluate them?
Sorry, which ligand are we talking about?
The SAMD9 ligand gene. The SAMD9
Oh,
ligand.
yeah. Not sure yet. Want to think
Is there a
about
functional
really.
test?
I don't have anything ready in my lab for that, but it would be a functional test really to look at whether or not it is gain of function really in terms of signal transduction.
Any other insights in the panel regarding SAMD9 ligand? Yeah, it's a gene causing MDS, and I think Marcin Wlodarski is in the US is someone who studies that, and Miriam Erlacher in Ulm, actually in Germany. Okay, so the next question is, why do you think somatic reversions occur? Any hypothesis for the same? And why do some DOC8, ADA2 more prone to it? So I think we discussed the DOC8 because it's a very telomeric, and therefore it's a hotspot for mutations. But I'm really surprised also with this somatic reversion, Roger. Who told the system to correct CD8 cells? Why? So who told that the CD8 cells need to revert rather than all the T cells?
Yeah, it's a good question, and we don't have the specific answer, but I assume it's like a selective pressure, So CD8 T cells are crucial to control Epstein-Barr virus. So because somatic reversion, you can have, for example, somatic reversion in a hematopoietic stem cell, but then you can only detect the effect in the cell that really has an advantage for this somatic reversion. So in this case, we don't know which is the origin. Obviously, it's not in a stem cell. I think we believe it's not in a stem cell, but probably a precursor of T cells. And in this case, CD8 T cells have expanded because they recognize the virus and they expanded due to the use.
I understand, but why don't we see at least some of it in CD4s as well?
Yeah, I don't know.
I see they don't have an advantage, but
it's
not a disadvantage.
Yeah, we don't have the answer. So it's it's a good question, and in this study we open a lot of questions that we cannot understand, but we don't know.
Then the question is, in the field of variable expressivity in inborn errors of immunity, what is the weight of methylation and or somatic reversion? Who wants to take that question?
I think the methylation will be quite, I mean, with the, grade 2 is a long-range sequencing. It's just the beginning of really trying to understand the effects of methylation of various gene expression. And as Dushan has shown in his allelic-specific paper, I guess this could be a mechanism that is responsible for some of the unusual allelic expression that we see in some of the immune-related genes. I just think there isn't, it's just going to be huge expansion in this area with just use of different sequencing techniques, with long-range sequencing now being used more frequently, where you have a methylation information readily available. It's still difficult to interpret properly.
Yeah. Thanks, Sinisa. So we are already 5 minutes over time. Unfortunately, there are still a couple of more questions in the Q&A session. Maybe the panelists can go there and respond to them in writing. This leaves me, Susa, for the closing the session, but without mentioning a couple of issues on behalf of ESID. The first thing is that I would like to remind you of the deadline to submit your abstract to our wonderful upcoming ESID meeting in Maastricht in October. Please submit your abstracts and your research to that meeting, and please register so that we can see each other all in person in Maastricht. Then I would like to advertise for people who haven't seen it that the ESID Genetics Working Party is offering a free genetic testing initiative. So if you have families where your healthcare or your country cannot pay for the genetic testing, and you do think that it changes the management of these patients or this family, please submit your application to the URL or to the QR code which is currently displayed. So we're looking forward to your submissions. There will be up to 50 tests being available per year for these families in need. It needs to be a physician who applies, so families cannot apply themselves. And the next advertising is for the collaborative ESID protocols. So ESID wishes to have a repository of research and diagnostic protocols. And this is all supported by the Educational Working Party. So that's a wonderful repository. Please submit your diagnostic and research protocols to that initiative. And last but not least, thank you for joining. It's been a wonderful seminar. Thanks again for all 4 speakers and talks. I learned a lot. It was really a lot of fun. Thanks for the lively discussions. Thank you for everyone who has submitted the questions. I think there's much more to learn in that field. So stay tuned and please come to Maastricht to discuss further issues. Thank you very much. Bye to everyone and thanks again. Muchas gracias.